My pleasure and honor to have here with us the President of the European Society of Hypertension, Professor Kreutz from Berlin, to talk us about the RAS inhibition and COVID.
Thank you very much, Vasilios. Professor Kreutz, personally, I'm very grateful for inviting me, but on behalf of ESH, I would like to thank you very much and congratulate you again for all the efforts and work to organize in difficult times such a very important successful meeting. So I'm very happy now as a continuation of our ESH secretary to talk about the renin-angiotensin system and inhibition in COVID-19. We are all aware it started very early in the pandemic that we were looking at reports, but also in the social media, we were overflooded with reports that there was a concern to use ACE inhibitors at the beginning and also later angiotensin receptor blockers in COVID-19. What's the reason or the background on this? We know this is an old system, very old slide I have also used always for teaching. This is the renin-angiotensin system and the way angiotensin receptor blockers and ACE inhibitors work. Very simple, the old system, and what is very well established for many, many years, that there are feedback loops. So renin definitely goes up very high, so it's not meaningful to measure renin in this, and that was also, we know that when we check for secondary hypertension aldo, primary aldo, always a discussion whether we should stop or not RAS inhibitors, but also the peptides are affected. So we know there are feedback loops in the renin-angiotensin system. We also know that the renin-angiotensin system, which is still a simple slide, is much more complex because we have in red the harmful red arm mediated by the angiotensin type 1 receptor, vasoconstriction, pro-oxidant, pro-inflammatory, pro-fibrotic mechanisms. But we have also on the left side in green, we have also a beneficial protecting, so a balancing arm. And there the ACE2 enzyme, which is another homologous ACE, it's a monocarboxylase, so it cleaves only one amino acid, is involved in activating actually this protective arm. We block the red arm, the harmful arm. Certainly angiotensin receptor blockers do not interfere with ACE, but ACE inhibitors also do not interfere with ACE2, do not interfere with ACE2. So ACE inhibitors do not bind or affect in any way directly ACE2. But if we block the renin-angiotensin system, we block the harmful red arm and we activate or further even activate the green arm, the protective arm. And we have also feedback loops discussed already on the other peptides such as angiotensin 1,7, etc. But I don't need to go into the details. So we have feedback loops, but angiotensin receptor blockers and certainly also not ACE inhibitors do not directly affect ACE2, but possibly by feedback mechanisms. In the lung, we have also renin-angiotensin system. We know in almost every organ, it seems that we have local tissue renin-angiotensin systems, not only plasmatic, but also in the lung, we have a renin-angiotensin system. We knew already before COVID in patients with bacterial pneumonia that RAS inhibition, particularly ACE inhibitors, might be protective because they, again, in the lung and the right side, they inhibit the harmful arm, while the protective arm, also protecting the lung, is indirectly supported by the inhibition, RAS inhibition. Now, the problem was, or is, that the ACE2, the homologous ACE, is the cell entry receptor for the virus, for SARS-CoV-2. Also for SARS virus, identified earlier already, and also for SARS-CoV-2. But very important, look at the small m, which indicates membrane-bound ACE, the protein bound at the membrane, not the soluble ACE, which is a different ACE that we can measure, for instance, in plasma or in the urine, which is important for our discussion. So the idea or the concept was at the beginning not really supported, but it was transported in the press, in social media, and in some good journals, even by letter to the editors, whatever, because they investigators or even non-experts, I should say, suggested that the use of RAS blockers can upregulate potentially ACE. If that would be true, then the infectivity would increase and the risk for COVID-19 would increase and the prognosis would be doubtful by using this. When we looked, we saw already from Andrzej Januszewic's presentations some data from our surveys that were guided by Thomas Weber and the Excellence Center. Many of you also participated in this. I'm very happy and grateful for this. We found that in the European Society of Hypertension Excellence Centers, 52 participated in the survey. They reported that in 27% of their patients, RAS blockers were stopped by the patients, or in 37% of centers, physicians stopped RAS blockers at the beginning of the pandemic because of this discussion. So we put up a task force to dealing with this problem. As you can imagine, it cost me really also and some colleagues 6 weeks between February, March, basically brain thinking and also headaches, dealing with this problem for many days in order to come up with a reasonable recommendation. So actually at the very beginning, the studies that reported an upregulation of ACE2 by using angiotensin receptor blockers or ACE inhibitors were animal studies, largely animal studies. And they measured, for instance, soluble ACE in urine. So if you use a RAS blocker, you can measure in the rat that soluble ACE in the urine goes up. But soluble ACE in the urine, for instance, comes from the proximal tubule, and that it's cleaved in the proximal tubule. You have the membrane-bound ACE, and you have a shed ACE that cleaves and transforms membrane-bound ACE into the soluble ACE, and that you can measure in the urine. Also, some experimental studies showed that the ACE messenger RNA in the cells, in the tissue, you can investigate tissue levels in the rat, for instance, were increased. But what was not shown in any study, we looked at this, and that on the protein level, at the membrane, you need for the cell virus entry, you need ACE at the membrane in the lung or the respiratory system. No study showed that in the lung or the respiratory system, a membrane-bound ACE would go up. So there was no effect. So the study supporting that there is an increased infectivity when using RAS blockers were very weak and not convincing, and therefore we couldn't wait for additional studies. So at the very beginning, very early, we concluded in the lung, the respiratory system, there's no upregulation of ACE2 and no risk for COVID-19. And we were actually among the first. Sometimes you see in the literature that the ESC recommendation is quoted or whatever I refer to, but we were among the very first on March 14 to suggest we should use RAS blockers as recommended. There's no risk. That was our idea based on the pharmacological, biological reasoning and study of the literature, but we didn't know clinically what happens. So therefore, there were many studies now, and if you look in PubMed, there are many, many studies, good studies, weak studies, and I just would summarize what we all know, or what is our perception right now. Certainly, we are very happy that Professor Mancia conducted in Lombardy region in Milan this important study, because Milan, Bergamo, and Milan were heavily affected with the first wave, and this is a match case control study 1 to 5. You see 6,272 COVID-19 patients, 30,000 matched controls without COVID, matched for sex and age and municipality of residence. And if you see here the odds ratio for being treated with an ACE inhibitor, angiotensin receptor blocker, mineralocorticoid receptor antagonist, to have a COVID-19, you see the odds ratio are close to 0 to 1 and are not significant. There's absolutely no difference between users and non-users of RAS blockers, but also calcium channel blocker, no effect, beta blockers, no effect, thiazide like diuretics, no effect. And there are many other studies, poor studies, high quality, large observational studies, that came to the same conclusion. If you look at one of the meta-analysis, the first meta-analysis, there are new meta-analysis. There will be a new meta-analysis published very soon, basically showing the same result, that all-cause mortality or occurrence of COVID-19 is not affected by the use of RAS blockers. But also the same applies to other first-line blood pressure lowering drugs, in summary. Only a few randomized control trials, all are very weak, very short looking, but very important, weak in terms of small numbers. The most first famous one was the BRACE Corona trial from Brazil, 659 patients. There's another one, the small trial, REPLACE trial. And there's even another trial, and there are some ongoing studies, maybe not conducted successfully, not reported yet. There's another small study with 100 patients using losartan. And these randomized control trials tested continuation versus discontinuation of RAS blockers, whether that had an impact on short-term prognosis, such as mortality after 30 days, absolutely also no effect. So it doesn't matter if you continue or discontinue a RAS blocker. So the conclusion would be to continue RAS blockers because we know they are protective for blood pressure control and cardiovascular protective and renal protective. So in summary, last year we concluded that using of first-line blood pressure lowering drugs has potentially no impact on acute effects of prognosis, death, mortality, and acute phase of COVID-19. But the limitations of these studies are also there. They are confined by the pathophysiology of complications of acute COVID-19 patients, hypotension, for instance, acute kidney injury. There are now new data that actually suggests that RAS blockade could protect against acute kidney injury in COVID-19 patients. On the other hand, if you have hypotension, the hemodynamic effects, also electrolyte effects, hyperkalemia come into play when we use RAS blockers. So there's a confounding of this. We have to be aware of this. When we looked onto, it's not one of my topics, actually, what is the role of blood pressure? Anjay discussed this already to some extent, and also the prognosis of hypertension, uncontrolled blood pressure. On the acute mortality, prognosis is unclear. At the very beginning, we thought that hypertension is a risk factor. If you fully adjust for hypertension, potentially, it's not a risk factor, an independent risk factor, but other comorbidities. But some of these comorbidities, such as heart failure, obviously also result potentially of hypertension. So whether or not hypertension is a problem, a risk factor, is not fully clear. I'm not really convinced that hypertension per se, uncomplicated hypertension, is a risk factor for COVID-19. So much more important is what we discussed already, the impact of blood pressure control, or the impact of the pandemic on blood pressure control and vice versa. Also, what's the role of the new variants we see now and in the future? What is the impact of vaccination? And also the treatments we use for COVID-19, like corticoids or other drugs that might impact also cardiovascular health and blood pressure control. So this is something for the future for research. Finally, some new aspects also in terms of research. We know that the cell entry apparatus for the SARS-CoV-2 virus is very complex. One thing is very clear. The ACE2 is the crucial entry receptor. But for the lung, you have different components that add to cofactors you need. For complications in the kidney, potentially infection in the heart, in the kidney, in the vasculature, in the brain, you have a repertoire of different cofactors that are necessary for cell entry and therefore for progression of the infection. For instance, the kidney, one early report showed in kidney cell line, not in humans and not in vivo. In the cell line, for instance, there's an interaction between vasopressin and the vasopressin receptor and the AT1 receptor. So basically the AT1 receptor in the kidney cells interacts with vasopressin and can promote the infection. So that is a hypothesis for the kidney, and thereby it could contribute to harmful effect in the kidney. A more recent paper in iScience, just published, they used organoids, not only cell lines, so they made kidney organoids by using stem cells. And this they analyzed the cofactors you need in addition to ACE2 in the kidney cells, and they could show actually infectivity of cells, proximal tubule cells in these organoids are mediated by angiotensin II and can be blocked by losartan. So we could protect the kidney with a angiotensin receptor blocker in terms of protecting against the SARS-CoV-2 infection of the kidney in complication. That's a hypothesis, is organoid studies. A new paper also published now with participation of many investigators from Berlin, from also our institution at the Charité. They showed, we know that there are potentially autoantibodies, we know that already, against the angiotensin type 1 receptor. And they showed that in patients with COVID-19, the levels of autoantibodies against AT1 receptors is increasing, and these autoantibodies are activating the AT1 receptor. So if you have autoantibodies against AT1 receptor, you can activate the renin-angiotensin system. The conclusion would be that an angiotensin receptor blocker could be beneficial in protecting against this effect. Finally, the genetics are also ongoing for more than 1 year. There is a COVID host genetics initiative. They combine a lot of data. There was an important first paper in New England Journal of Medicine in Nature. Very recently, in March now, papers in Nature Genetics. They are altogether up to now apparently 23 different loci on the human genome that have an impact on COVID-19 infection. But the very recent paper by Horowitz now confirmed also again that the ACE2 gene, there's a variant that affects ACE2 expression and can protect against infection. And that works together with another locus on chromosome 3, and they are potentially interacting. So this is very interesting, and potentially for the future, also maybe for treatment effects. This variant, the ACE2 gene, is a low frequency variant. The frequency in the population is about only between up to maybe 2%. So there are 2%, let's say 2% of the population, our population, have a variant in the ACE2 gene. That they could show this in this paper in the liver at least, and they measured the liver expression in the variant. And if you are a carrier of the variant, the liver expression of ACE2 is reduced significantly by about 40%. And that fits very well with the risk for the, actually by maybe chance or plausibility, the risk to obtain a COVID-19 infection in those patients who have this variant is lower, 40% lower. The odds ratio you see at the bottom is 0.6. So we wonder why some patients, probably also younger patients, are protected, and that could be due to variants. And they even calculated the genetic risk by combining all these factors together. And that could be maybe something helpful for the future in terms of also predicting risk, depending on the pandemic and the new variants we get. So this is one study we would like to see and support. Is there a difference between antihypertensive drugs in terms of prognosis if we use them? This is a small study, only 566 patients, again from Italy, showing that if you use angiotensin receptor blockers, mortality is reduced as compared to ACE inhibitors or other blood pressure lowering drugs. But it's a very small study. We have now a study we will present at the Athens meeting. I'm participating in the study 10 times as much, very careful characters, patients, number of patients, more than 5,000 patients. We did not find this. So it's also a question of number and quality of the studies. And I think that's my final slide. A very nice review. I encourage you to read by Professor Corrao and Professor Mancha, looking at the positioning and the very the high importance of real-life research, observational studies, because we do not have randomized control trials in this pandemic. But the quality of the studies and the number of patients and how you adjust is very important. And therefore, we need more studies now also in the future with hypotheses that needs to be investigated. And I think this is very important. Up to now, I think at the very end, this is my very last slide. I think I showed this slide already in April 2020, and many colleagues showed it. And I think it's the same slide I can show today, that the treatment algorithm we discussed and also Antonio Coca very nicely presented and Jana as well, and the other colleagues, should be followed. And I would go even further. It should not only be followed, but it should be even intensified, because we know the patients, for several reasons, as we discussed also with Andrzej Januszewicz, the risk to have poor blood pressure control and to be also protected through the pandemic is more important now, and therefore nothing to be changed in terms of, and certainly RAS inhibition is still the backbone of therapy and very important, not only for hypertension, but also for other diseases that we discussed during the symposium. Evaristo, thank you very much.
Thank you.